hear me now? Yeah, there we go. Oh, I will, yes, definitely I will. But these are questions that are basic questions that are foundation questions. So if you're not, if you're not studying it, the material beforehand, then it's going to be a problem because you're going to have so much material to deal with before the exam. And I think that's one thing that I'm seeing and noticing from students. They're not studying in advance. And you've got to push to the extent where you get that foundation early so that you can build on it. This course is going to be heavy, but it's interesting. I asked you last time at school, let me rephrase that question. How many of you are going to healthcare profession? Okay. So we're going to be talking about a lot about diseases. All right? And if you don't, and I'm going to ask you questions on diseases. And if you don't know the how it's happening, then you need to come to me and ask me how it's happening. Because that's really the mechanism of the part of the diseases that are so interesting. Not just the functional consequence, I throw in the drug, I get this, great. But I want to know, is it going to affect me in other ways? That process of thinking comes here. Okay, so that's the most important thing. So those are questions. If I talk about a disease, which I'm going to talk about diseases today, and if that book doesn't have as much material in it, I supplement it. The book is good because it's short, and you'll be able to read it pretty quickly before coming to class. And you've got to spend the time. If you have a course, and I know you probably are taking about 15 credit hours, among other things, work and everything, but somehow you have to squeeze it in. So, number one, I hope that you were kidding me when you said you didn't know how many chains there are.
We have different papers.
What paper? This question. This is what I wanted you to do. This is the question that I wanted you to do. That sheet we're going to do afterwards. Is this not what you guys were working on? Yeah? Okay. All right. So can somebody tell me who knows the answer to how many chains there are in hemoglobin? Yes.
2 alpha and 2 beta.
Okay. So 4. Right? That's like basic question of what a hemoglobin is. Alpha 2, beta 2. This is important for today because we're going to talk about hemoglobin, the fetal hemoglobin. It's not alpha 2, beta 2, it's alpha 2, gamma 2. And that's important. All right, so the second question, the number of heme iron complexes are, how many do you think there are? If there are 4 chains, then how many are there? Yes? 4, right? So remember those little pink things that I showed you with the iron embedded within each of those alpha and beta? There are 4 of them. Molecular oxygen binds to what? What does it bind to? Yes. Correct. Distal histidine in the ferrous ion. So distal histidine stabilizes the ferrous ion. It's not ferric ion, it's ferrous ion. Fe plus 2. It's in that form, Fe plus 2. And remember what we talked about, structural features. And I went through it with different slides. So iron is going to bind to 6 states. Okay, the 4 of them comes binding to the pyro rings, and 1 of them comes to binding to the proximal histidine, and the other one comes from binding to the oxygen. You've got to know what it binds to. And to end it off with this, distal histidine. What is one, at least give me one function of distal histidine we talked about. Yes? Prevent carbon monoxide from binding. Yes. Prevent carbon monoxide from binding so that we prevent anything that has to do with overexposure to carbon monoxide from poisoning the system. Okay, so these are questions, these were things that we went through extensively last class, even before the peptide sequence, even after the peptide sequence. So if you're not getting these types of questions, these are foundation. I haven't even built on them yet. I'm not even giving you applications yet. If you don't know the foundation, because that's where we started, foundation, you're going to lose points on this. Definitions is what I'm saying. You guys are doing it in the assignment, but then, there's so many resources out there. You can just type it in and you get it, all right? But you're overlooking them. It's not enough to know a definition, you have to apply them. If you don't, the first thing is knowing the definition. I've seen a couple of you this week who got the question right on an assignment of what an amphipathic molecule is, but then it's not being retained. It's not showing up on the exam, okay? So the first thing is, obviously, to know definitions. That's the first thing you need to know, definitions, and then be able to apply them. There'll be little definitions. I gotta tell you, there's little definitions. I've given you 37 questions, and the questions will have the words, the scientific word in it, and you have to take that scientific term, knowing the definition, and be able to apply it. But even before some of you get there, you need to know the definitions and the basic things. Okay, if you need to print your slides out, that's what I would strongly recommend. Don't miss a class. Okay, and come and see your exam. You need to come and see your exam, okay? I'm not gonna judge you. It's your first exam. I've seen it over 13 years. It's not something that's new that I'm seeing, but it will build, okay? So I encourage you to come and look at your exam, see what your weaknesses are and your strengths are. Change your study habits, because it's not gonna be the same as you've done it before. Some people are visual learners. Some people need to write everything down. I'm not going to write everything down because that's where the book comes in. Okay, I'm going to write some things down for you guys, but then, depending on what you need on there, you need to put your notes down. Okay, so the first thing that you learned today is go home and study from last lecture. All right, so I do need to keep going, because this is very important. Hemoglobin is very important, and I don't think you escape it. When you go, for most of you, or 90% of you who are going to healthcare professions, or even grad school, you're going to use these concepts of analyzing data, looking at graphs, and synthesizing it, and coming up with what the data means. And some of that we're going to do today with hemoglobin. Among things that you guys are also interested in, which is also is sickle cell disease. All right, so last class, let me go back to last class and review a little bit of what's coming. So we did all of this. All of this was all about the features, right? And there's different ways that I taught you that. Where were we? Okay, so here, I do want to go through some of these terms again and review, because I feel like I do need to review some of them, because it's important. All right, T state, R state. Right? We talked about T and R. T can be used interchangeably as a deoxy state. There is no oxygen. Maybe there's 1, 2, but still it's considered T state. Because remember, there's 4 sites. When it's fully oxygenated, that means all 4 are bound with oxygen, then it's an oxy state, or we consider that an R state. So this is your first example, like I told you guys last time, your first example of where you're going to see that T to R transition with hemoglobin. This is not the only one. There's so many different proteins that toggle between T and R. It's like a light bulb switch, right? We're not going to make synthesis and make proteins all the time because it needs energy. So we're going to have to regulate things. We're going to have to shut things off and turn the other part on, and vice versa. And this is where the T to the R state comes in. Okay, we talked about this already. told you that myoglobin is hyperbolic. Hemoglobin is sigmoidal. S-shaped, sigmoidal, hyperbolic. And whether oxygen binds tightly or not, whether it's going to be the T state or not, the curve is going to shift. Okay, and that's what we're going to be working on today. How the curve is going to shift based on the effectors that are going to bind to hemoglobin. Told you the importance of 100 torr, using the 100 torr in the lung versus 20 to 30 torrs in the tissue. All right, that's important today. It's important to highlight that because we're going to be using that. So when we look at this, I also told you, notice why hemoglobin is considered to be the protein of choice. Because right now we're not really specifically looking at values, but we will be in a minute. Notice that with the hemoglobin, 66% of oxygen is being released to the tissues, compared to 7%. Right? So if 66% is being released, that means more oxygen is going to the tissues. It's not holding it as tightly, but it is not releasing everything. And that's what you want when you transport protein of oxygen. You don't want something that binds so tightly that it doesn't release, but you don't want something that releases everything either. I'm going to come back to this and how to look at this, but I showed you guys how to do this last time, but I'm going to review it because we're going to be looking at it more specifically. When we talk about hemoglobin, the fact that it is going from T to R, and it is sigmoidal, these are features of allosteric proteins, right? There are some features of allosteric proteins, and one of them is that it goes from the T to the R, and it's sigmoidal in shape. It's going to be more complex. The complexity will come in next semester when we actually compare allosteric enzymes with another type of enzyme, Michaelis-Menten. Another important thing that you need to know about allosteric is its positive cooperativity. So that's another feature. So positive cooperativity means that when one oxygen binds, there's sort of a communication. There's going to be a conformational change that allows another oxygen to bind. More conformational changes, another one binds, and another one, until all the pockets of that hemoglobin are filled with oxygen. So that's positive cooperativity. So this is what we're going to talk about today, the allosteric effectors, and there's three. Okay, so we talked about the allosteric, so here's another feature, quaternary structure. So there's primary, there's secondary, tertiary, and quaternary. So all allosteric enzymes will have quaternary structures. And all allosteric enzymes will have stimulators or activators. Right? So jumbled up in your notes are all 5 or 6 features that we've talked about that has to do with allosteric enzymes. They will have the T to the R state. They will be sigmoidal in shape, not hyperbolic. They will have positive cooperativity. They are complex in activity. They possess, like we talked about before exam 1, they possess quaternary structure. And today specifically, we're going to talk about those allosteric effectors. Okay, so these allosteric regulators. Okay, so you've got about 6 different features for allosteric enzymes. So we talked about cooperativity. What is the definition of cooperativity? Since we're talking about hemoglobin, so when one oxygen binds, another one will bind, and so forth. That's cooperation, so that's cooperative binding. So in with the example of hemoglobin, if it's all the same ligand, everybody knows the term ligand, so in this case we're looking at oxygen, and it's the same molecule that's binding, it's called a homotropic effect. when they're different, we're not talking about different ones this semester, but when they're different ligands binding, they're heterotrophic. So we talked about this. So this is where we're going to start with today, how to make that conversion from the T to the R state and vice versa, and how the graph will shift. That is the most critical thing as far as looking at graphs and analysis. And the last exam you saw a lot of graphs, the titrations. And so you'll see those types of graphs with hemoglobin. There are two types of models that one has envisioned. One is a concerted model, and one is a sequential model. So with the concerted model, this more has to do with probability. It's really hard to say which one occurs because it's so fast. So with the concerted model, the researchers have said the default is predominant 99% is T. That's what they are predicting. 99% of the state that we have seen is in the T state. And that converts over to the R state. So it all has to do with probability. So the default is the T. Sequential model, I like this sequential model. See, this one is your concerted, where it's either T or R, and they've seen predominantly T state that's converted into R. Sequential model is conformational. And that has really to do with cooperativity as well. Both of them have to do with cooperativity. It's just which one have you seen in an experiment? So sequential model is a more conformational. That means, like I said, if one binds, you see some conformational changes that allows another one to bind. More changes, and then another one binds. And more changes, the protein changes, and another one binds. So that's a sequential. Okay, but either case, we're still going from T to R. See? Any model you see going from T to R. You can believe whichever one you want, and so because they could not figure out how this two, whether this or that model occurs, they kind of merged it together, and they came up with this model where they say you see more of the T. They're not saying percentage-wise, default. You see more of the T. And in this area here in green, when oxygen binds, it begins to convert over. And remember I said that in the model, you can have all T states with one bound or two bound. In this model, they're trying to do a compromise and saying that you can have one bound, it will still be considered a T state, but as it moves, you're starting to see a change, conformational change. And so when 2 binds or 3 binds, notice these changes are occurring, and they call that the R state. So you have some already in the T state. They're not saying it's all 99%. They have some in the T state as a default. And then as you're moving, as soon as at least 2 binds, they call that the R state due to conformational changes. Okay, so that's where the fine line is, because it's really hard to catch something that's so quick. Okay, so the binding of myoglobin, I've already told you, right? It's not sigmoidal. You can look at the shape. You've seen this shape, I'm sure, many times in a math class, and it's a hyperbolic curve. One of the things that we need to understand is the term P50. So P50, the definition of that is the partial pressure of oxygen that's required to give 50% saturation. So that means 2 out of the 4 binding sites of hemoglobin are bound by oxygen. So that's 50% bound by oxygen. Make sense? Okay, so that's the value that you're trying to find, the partial pressure of oxygen, where this particular protein is bound to its ligand 50%. So the way we do that is we look at our y-axis, which is saturation. Saturation means that all of the hemoglobin is bound, right? Or in this case, myoglobin is what we're talking about. So all of the myoglobin is bound by oxygen. And at 50%, right here, you bring it down, and that value right there is your P50. That particular pressure of 2.8 torr, the myoglobin is bound 50% of the oxygen. So that means if you have a low P50, right here, if you have a low P50, that means the affinity for that oxygen is going to be very tight. We're going to look at that difference between myoglobin and hemoglobin in a little bit. Okay, so just keep that in mind, the back of your mind. Low P50 means it's tighter binding. Everybody's good with the word affinity, tight binding? Okay, so affinity means binding. So this is binding. You do the same thing, right? In this graph here, you look at the P50 myoglobin, and you look at the P50 in hemoglobin. And both of them always look at where it's saturating. My graph doesn't necessarily have to be at 100%, it may be at 50%. Okay, so don't always assume that it's at 100%. Look at the graph, look at your y-axis, look at your x-axis. So y-axis tells me that both hemoglobin and myoglobin are reaching at 100% saturation. And in this case, I'm just going to look at P50. And so with the P50, with myoglobin, you go to the 50% mark and bring it down. It's about 2.8. The same thing, 50% with hemoglobin, you bring it down, it's 26. What did I tell you? The lower the P50, the higher the affinity. And that makes sense. That's what I just told you. Because remember, 66% of oxygen is getting released to hemoglobin, so it's not binding as tightly as myoglobin. So these numbers need to make sense, right? If you're releasing 66% oxygen, then your P50 is going to be high. If you're getting the reverse answers, you're doing something wrong, right? Things need to make sense. All right, so here, I just showed you, about 66% oxygen is being released. And myoglobin, we saw about 7%, I think, was released. Right? Do you guys want me to go back here? This is where it's coming from. Those numbers. Okay? So when you look at that, it makes sense. If you're releasing more oxygen, it's not binding as tightly, which means your P50, the personal pressure of oxygen that it binds to 50% is going to be much higher. Okay, so that's why you can rationalize with this number that 26 torr means it's lower binding, more release of oxygen, and that's why hemoglobin is considered the protein of choice. It's not binding too tightly, but it's still able to release. All right, I'm just going to go through just the top one right here, this one right here. I don't need to go through all the scenarios. Just to show you that it's important for this curve to be sigmoidal. So both of these proteins, the transport protein in our case is hemoglobin, it's reaching at 100%, right? It's binding tightly, but it's also releasing. If you have a curve as a hyperbolic, as I just showed you, it's not going to release as much. It's going to release about 7%. And that's just not enough. Okay, so that's why we want to have a curve something like this. Do you guys have questions about this part of things? We're going to dive into more about looking at calculating the P50 and the oxygen unloading by looking at graphs. All right, let's look at what we're going to look at. I already told you hemoglobin is an allosteric enzyme. So that means it has allosteric effectors. Allosteric effectors are those effectors, stimulators, or inhibitors that bind to a protein, any protein, because in this case we're looking at hemoglobin. Next semester we'll be looking at different proteins that are allosteric effectors. So they bind, but they're not binding to the oxygen binding site. So they have a site separate of its own. Okay? So let me just make something cartoon here. This is for visual learners. Let's say these are my hemoglobin. And say, here's my heme, binding to oxygen, right? You guys remember where the oxygen binds with coordination site? Okay. Okay, 6th coordination site. It binds to, right? The oxygen binds to that 6th coordination site. The oxygen is here and bound to the heme. These allosteric effectors that we're going to talk about do not bind to the 6th coordination site. They have a site somewhere on the zone. Okay? Somewhere else. On the hemoglobin that it binds to. So they are completely separate. They do not bind to the 6th coordination site. Okay, so that's the key. So using these, we're going to talk about 3, and I'm going to go through it. First, how it binds, and then how it affects, functional consequence. How does it affect the oxygen unloading and the P50 value? So 2,3-BPG, hydrogen ion, and the CO2, carbon dioxide. Those are the 3 effectors that you're responsible for. How is it binding to hemoglobin? And what does it do to oxygen unloading with reference to also the P50? Because they go hand in hand. We're also going to look at adult hemoglobin and fetal globin. That's the question I asked you guys. How many chains are there? There are 4 chains. Adult hemoglobin and fetal globin will also have 4 chains. But instead of the alpha 2, beta 2, the fetal hemoglobin will have alpha 2 and gamma 2. And this is really interesting. For those of you guys who like developmental biology, you'll see an interesting trend with the expression of these different genes. And then, of course, you need to know the disease cell, sickle cell anemia. And that has to do with amino acids. Okay, so these are the 3, like I said, that we're going to go through one at a time. First, with the 2,3-BPG. So they took a pure hemoglobin, right? Just a pure hemoglobin. They purified it, just a pure hemoglobin, versus like a blood sample of hemoglobin. And what they saw was, with the hemoglobin, what shape is this? They saw a sigmoidal shape. Okay? When they took it out, the pure hemoglobin, they purified it, and they wanted to do an experiment with it. What they saw was this shape. So what does this shape look like to you? The black. Hyperbolic. It's starting to look like a myoglobin. Right? So this is your sigmoidal. And this is starting to look like a myoglobin, the hyperbolic curve. Why? What happened? What is in the system, in the blood system, that is making hemoglobin release oxygen at 66%, versus in a test tube where you purify hemoglobin and you see only 7%? What is that something? And that something is 2,3-BPG. Okay, so that's your first allosteric effector. So now we're going to look at what is this feature in the 2,3-BPG that's causing this release. And that's the toggle between the T and the R state. Okay, so let's look at this 2,3-BPG structure. 2,3-BPG is anionic, meaning it has negative charges. There are your negative charges. So it's all negative. So when you think back to your amino acids, there's some sort of an interaction that's occurring. Right? Some sort of interaction is occurring with a negative charge with amino acids that are suddenly positive charge within the hemoglobin. They're causing it to convert from a T to an R state. That's the hypothesis. That's what we're envisioning. Something is happening, the 2,3-BPG levels are there in the blood that's making this hemoglobin release more oxygen to 66%. And so they looked at the quaternary structure, going back to their primary structure, and I kind of do a blast, and I showed you some of that last time, where a whale versus a human, and we saw some consensus sequences, right? Sequences that were there in both. And one of the things that were there was obviously histidine. I told you, because it is a protein, this particular histidine, is located in the inside away from the water. And that's unique, but there is a function, right? The proximal histidine and the distal histidine. That's where that came from. But they're secluded and isolated from the water. So when they looked at the states, here's my alpha 2, here's my beta 2 here, beta 1, beta 2 here. So these structures, they saw these lysines and histidines. And you know that lysines and histidines are positively charged, right? They're basic amino acids, right? So these basic amino acids are present and exposed in the T state. Okay, so 2,3-BPG binds to the T state of the hemoglobin, and when it binds to the T state of hemoglobin, oxygen is going to get released. It stabilizes the T state, which is the deoxy state, which means oxygen is getting released.
Can you say that again?
Yeah, so 2,3-BPG being anionic, right, has negative charges, binds to these positively charged amino acids. When they bind to it, these positively charged amino acids are exposed in the T state. Because remember, you have the T and the R. So the T state is exposed, right? Conformational changes occur between the T and the R. The same amino acids are there in the R state, but remember the folding is different between the T and the R. So in the T state, these positively charged groups are exposed. That allows binding for the 2,3-BPG, right? Charge to charge interaction. And when they bind, it stabilizes this T state. So in the end of all of this, I will be summarizing. So it stabilizes the T state. So when it stabilizes the T state, it means it's deoxy state, and so more oxygen is being unloaded to the tissues. Does that make sense? So that's what's happening here. When you have in this graph 2,3-BPG in the serum, in the red blood cells, the presence of 2,3-BPG binds to the T state, stabilizes it so that more oxygen gets released. Okay? So here's my binding pocket here. Remember I told you it's not binding to the 6 coordination site. It's binding somewhere within the hemoglobin. It has a site of its own. And so 2,3-BPG, here's my 2,3-BPG. This is my 2,3-BPG, right there in the middle. So it's binding to some pocket within the deoxy state where it binds to all those positive charges and stabilizes it. And more oxygen is getting released. Okay, so that's the mechanism. That's how it happens. I'm going to take a little bit of a detour and talk about fetal hemoglobin and adult hemoglobin, because we have to reference that in terms of the 2,3-BPG as well. Okay, so for those who are interested in developmental biology, this is really interesting. I don't even do developmental biology, but it's always interesting to me to see that how gene expression is between the stage 1 to all the way to stage 2, from fetus to all the way to adult. Okay, so the black bar here is alpha, before birth, after birth. Before birth or after birth, look at that expression level of alpha. Okay, so y-axis is your total globin synthesis, and gamma alpha is saturated, right? It's pretty much high levels in both adults and fetus. Then you look at beta. Look at the levels of beta before birth. It's very low. Don't worry about the other ones. These are different isomers, but beta is very low. After the baby's born, the levels of beta go up. So something is regulating this system where you have low levels of beta. We don't want beta in a fetus. Why, I'll explain. But then after birth, the beta starts to increase. But the fetus still has 4 chains. It just doesn't have beta. It has small, low expression levels of beta. it has another isoform called gamma. So babies before birth, alpha 2, gamma 2. Okay? The fetus is going to be alpha 2, gamma 2. As an adult, as soon as the babies are born, the gamma expression begins to go down. It's not zero. Notice it's not zero. It's just turned off. The gene is still there. We still have this gene present. We have gamma, and we have these genes still present. It's just off. And then alpha, or beta, begins to go up. Okay, so it's alpha 2, beta 2 as an adult, alpha 2, gamma 2 in the fetus. Yes?
So, I noticed on the graph that sigma appears after birth. Is that, do you know anything about that?
This one? Which one are you talking about?
The bottom. Post-birth, right near the bottom, is sigma.
This one?
Yeah.
That's delta?
That's delta.
Delta, yeah. So delta, yeah, we don't know what that is, but the hemoglobin has different isoforms, but it doesn't turn on that we have seen. So when I talk about thalassemia, which is a disease of a disorder where the adults don't have the alpha or the beta, the gamma seems to pick up to compensate for the loss of the alpha or the beta. These ones, there is very low expression, but it doesn't do much. As far as we've seen with diseases, it doesn't pick up, the expression doesn't pick up. Okay, so we're going to talk about, do you guys have any other questions about this? All right, so what is it that makes, that the baby needs, the fetus needs a gamma instead of a beta? So remember where the sites are within the beta chain? We've got some histidines and we've got some lysines, right? In the beta subunit. Right? So within the beta subunit, we've got all these positively charged groups. So we're going to convert the baby, we're going to convert it. So in the fetus, the gamma still has some of it, but the gene itself, okay, it's a 80% beta gene, which means that structure-wise, primary sequence, 80% of the amino acids are the same. But there are some slight differences, and that difference comes really from the histidine groups. The ones that I just showed you in the pockets, that histidine is actually a serine within the gamma. Okay? And that's important because we're losing charge. So that means the oxygen is not going to, the 2,3-BPG is not going to bind as tightly anymore to that binding pocket, which means it's going to stabilize the R state. Remember, the fetus is dependent on the mother for the oxygen, right? So it's not going to release all of its 66%. It's going to be a little less than 60. It's going to be a little bit more than 66%. Okay, so the oxygen is going to bind more tightly to the fetal hemoglobin, stabilize more of the R state than the T state. So when you're stabilizing more of the R state, it's going to retain that oxygen. So instead of 66%, it might be 77%, right? Or a little bit less, 50% or so. We'll look at that in a little bit. So let's just look at the loss of histidine. So numbers are not important. One of the histidines that I just showed you at location 143 of that chain is, I wouldn't say lost, but it has been replaced within the genome, right? That's gamma. Instead of the histidine, it's a serine. So the serine, remember, has an OH group, so it's not a positively charged, it's not basic, right? So we're losing that charge. So when you lose that charge, that 2,3-BPG, which is anionic and negatively charged, is not going to bind tightly because you've lost that histidine. And more oxygen is not going to be released. Okay? So how is it going to affect that sigmoidal shape in the fetal hemoglobin? So here's your maternal. Okay, so the maternal red blood cells is, we can say that's the adult, so about 66% is being released. Right? So how do we calculate that 66% release? We looked at the lungs. So lungs is 100 total. And so this might be 20 or 30. So at 20 or 30 is at this point right there, and this point right there. and you take the difference. Okay, that's what we did last class. We took the difference. And we said from 1, and we look at the maternal. I'm just going to make it 0.34 roughly because I want to make it 66%. So 1 minus a 0.34 gives me 0.66 or 66%. So that's what we calculated last class. So then when we look at the fetus now, you do the same thing. How much oxygen is getting unloaded? All right, so again, if you look at the fetus, it's 1. Let's just say it's 0.55. All right, so that's point right there, 0.55, 0.45. So about 45% is being released. So that makes sense because the 2,3-BPG is, the concentrations are going to be there, right? That's not going to change. The concentration is there. So it's not manipulating any of the concentrations of the 2,3-BPG. Okay? It all has to do with the gene of the gamma. So because of the change in the gamma from the histidine to serine, that 2,3-BPG is not going to bind as tightly, which means it's not going to release the oxygen. The T state is not stabilized, but the R state is. So it's going to be the oxygenated version compared to the adult. Is that good? Do you have any questions about this part of calculating? Yes?
So why are you using the points for like the 0.34 and the 0.45?
Okay, so remember what I told you guys, this number is important. This is the partial pressure of oxygen in the lungs. So 20 or 30 is the partial pressure of oxygen in the tissues. Right, so those numbers are important. So we go from the lungs. The lung carries the oxygen to the tissues and it's unloading. Right, the lungs are carrying the hemoglobin that's oxygenated, is going to carry from the lungs towards the tissue and it's going to unload. So in the lungs, that particular hemoglobin is at a point, is at a 1. 1 point, right there. Okay? And then you take the point that it intersects and you go up, and with the maternal hemoglobin, it's about this point, and I'm roughly estimating this point to be about 0.34. This point right there, that it intersects is 0.34. And that's where I'm getting the oxygen unloading. So the oxygen that's getting unloaded here is from this point to this point, and that's your oxygen unloading right there. And the oxygen that's getting unloaded from the fetus is from this point to this point. So I'm just roughly estimating what the values are. But that's how you calculate your oxygen. So let me ask you guys a question. Did I put that question? Okay, let me ask you guys the question. So knowing that you're releasing more oxygen to the tissues in the adult than the fetus, what would the P50 look like? Would it be higher in the fetus or would it be higher in the adult?
Adult.
Okay. Right. So the affinity P50, remember, is the partial pressure at which 50% is saturated. So with the maternal red blood cells, more oxygen is being released. So the binding affinity is less. So P50 is going to be much higher. Remember, it's reciprocal. Higher the P50, the lower the affinity. So the shift here will be more towards the left. And you can prove it to yourself, right? You don't need to memorize the concept that it has higher P50, the lower the affinity. You can prove it to yourself by looking at the graph. So let's go back and let me see if I can get another graph here. No, this is the other one. Okay, that's okay. So let me use another color here. All right, so let's look at the P50. What did I tell you about the P50? It's 50% saturation. So with the maternal here, 100%, and then you go to 50%, right? And so at P50, for the adult, this may be about, I'll use the same value that they used before, it's about 26 torr. Okay, so the point right there, at 50%. And then you do the same thing with your fetus. You bring it down, it's a little bit less. So the P50 of the fetus is I don't know, about 20 maybe? I'm just roughly guessing at this point. Your graphs will be better. You'll be able to figure it out a little bit closer, okay? Okay, so that makes sense, right? That makes sense. So you've proved to yourself that, yes, my P50 is going to be much higher in the adult than in the fetus by looking at the graph. And that's what you're going to have to do. You're going to have to come up with the P50, and you're going to have to come up with the oxygen unloading. And we're going to do examples of that with our next worksheet. Okay, so this is really the effect due to the loss or the replacement of the histidine with the serine. 2,3-BPG doesn't bind as tightly, and so the R state is stabilized unless oxygen gets released to the tissues. So that's a summary of that. Okay, with high altitudes, what happens? As you're climbing up a mountain, the concentrations of the 2,3-BPG is going to change. Because as you're climbing up elevation above sea level, you're going to have to release more oxygen to the tissues to compensate for that elevation. So in your body, the genes are not going to change, right? You're not going to mutate your alpha or beta to compensate for this. So what's going to happen is your 2,3-BPG levels in your body are going to change. So 2,3-BPG is going to rise so that more oxygen gets released. Okay? So as 2,3-BPG begins to rise, so here's your sea level state, above sea level. Notice what's happening as 2,3-BPG begins to rise. It's going to stabilize which state? T state, good. Right, it's going to stabilize that T state. So that means what? Is more oxygen going to get released or less oxygen going to get released? More, good. So more oxygen is going to get released when it stabilizes T state, and that's what you want. As you go up higher levels, altitude, that increase is going to be due to compensated by increasing your 2,3-BPG levels. Then after you come down, your 2,3-BPG levels will go down back to normal. Right? So there has to be a way to release more oxygen. So by increasing 2,3-BPG, you're going to have it bind that T state, and so more oxygen is going to get released. So what does this mean in terms of P50? So if more oxygen is getting released, it's not binding as tightly, right? So your P50 is going to start to rise. So higher the P50, the lower the affinity. Does that make sense? You guys have any questions? So, yeah, there's my higher P50. So it has to make sense, right? If more oxygen is getting released, P50 is going to start to rise. Right? You're going to have to see more of a rise in P50 because that's going to be the partial pressure at which you're seeing 50% bound. And you need more pressure for it to bind to the oxygen. Okay, so increase in P3-BPG is what compensates the body to release more oxygen to the tissues. And so more of the unloading will happen. So when you look at a graph, so this is, say this is normal level, and as you go up high altitude, so let's just say above sea level here, so there's going to be an increase in 2,3-BPGs. As 2,3-BPG increases, there's going to be more oxygen unloading, and the curve, the sigmoidal curve, will shift more towards the right. Right? Just like you saw with the fetal hemoglobin, because it's retaining it, the shift was more towards the left compared to the adult. And when you see it with the high altitudes, there's going to be an increase in 2,3-BPG. Okay, but any case scenario, when there is a 2,3-BPG rising, the shift is going to be towards the right because more oxygen is going to get released and the P50 is going to start to rise. So the second and third as well of the allosteric effectors is what happens when there's an increase in hydrogen ion concentration. Right? So this can be written in terms of hydrogen ions or pH, right? We can reference it as either. That and carbon dioxide. So what happens? Okay, so a good example would be when you're exercising, there's going to be a rise in that hydrogen ion concentration and CO2 is going to be exhaled out. So when that happens, you're going to have to see an increase of oxygen going to your tissues. And more aerobic respiration has to occur because you're going to have to get that energy while you're doing strenuous exercise. One of the things we started earlier on was the carbon dioxide combining with the water to give you this carbonic acid and bicarbonate buffering system, right? This was an important buffering system. And we said as the hydrogen ion concentration increases, we're going to push this reaction towards the left so that we can expire out the CO2. And so that was the importance of this buffering system that we have seen. So how is it related to hemoglobin? So let's look at what the pH or the hydrogen ions do. So when the pH decreases, what does that mean? Hydrogen ions are increasing. Right? When pH decreases, hydrogen ion increases. So as pH decreases and hydrogen ion concentration increases, more oxygen is going to be unloaded to the tissues. Right? So when you're exercising, there's going to be that release of that hydrogen ions. And so that body is going to compensate by releasing more oxygen to the tissues. And so I'm showing you the graph first here. I'm going to talk about how in a minute. But notice in this graph, so as the hydrogen ion concentration increases, you're releasing 77%, 10% more oxygen to the tissues than in the normal state. And that's what you want to see. Again, this is another allosteric effector, which means it's not going to bind to the sixth coordination site. It has a site of its own that it binds to, undergoes that conformational change to get it more stabilized to a T state. So as the pH, I did say that, okay, so the T state is stabilized. So if you increase your hydrogen ion or decrease the pH, you're going to stabilize the T state, more oxygen is going to be released, and when you see a high P50 or a low P50, as you push more towards the right. High P50. Right? As pH decreases. Or hydrogen ion concentration increases. Either way, right? You guys get that, right? pH decrease or increase in hydrogen ion concentrations, your P50 is going to rise because it's not going to bind as tightly anymore, and so P50 is going to increase. So you're going to have to toggle back and forth between understanding the relationship between P50 and oxygen unloading, how to analyze these graphs, come up with a P50 value, rationalize how much oxygen is going to get unloaded. So how does this happen? Let's take a scenario where let's take this case scenario first, with a pH of 7.2, which is below the physiological range. So what happens when you have a higher hydrogen ion concentration is one of these histidines in the beta chain gets protonated. And when it gets protonated, it's going to stabilize the T state. Okay? So when it stabilizes the T state, more oxygen gets unloaded. So that's the mechanism. So just through having that protonation, you're going to have that salt bridges, which includes, again, the hydrogens added to the imidazole ring, which then hydrogen bonds with one of the other amino acids, so there's your hydrogen bonding as well as your ionic interaction between the positive and the negative charge. And so that stabilizes the T state. So always think, okay, there's a T and an R state. In the T state, these are exposed. And so when it's exposed, it's going to protonate that imidazole ring, create that salt bridge interaction. And when it does that, you're going to stabilize the T state and more oxygen gets released. So that's the how it does it. Okay, so this is the functional consequence of having higher levels of hydrogen ion concentrations. What happens? Right? Your P50 rises, more oxygen gets unloaded. This is the how. How does it do it? Okay, so we've answered the how question and we've answered the functional questions of what happens. Okay, so this is just a summary of what I just said. So as the pH gets lower, the histidine gets protonated and stabilizes the state. Which state does it stabilize? It stabilizes the T state. And the reverse is true. As pH rises, that R state is stabilized. So if you look back and you have a pH, and I think I have a graph like that. If the pH is at 7.6, the R state is stabilized. Less oxygen is going to get released. Okay, so that's how the graph will be, from higher pH to all the way to lower pH. Okay, so there it is, okay. So here's my graph. We know this is going to be our myoglobin because it's hyperbolic. We know that as the pH decreases, the shift is going to be more towards the right because more oxygen is going to get released because the T state is stabilized. The last one is carbon dioxide. What does that do? It too goes hand in hand, right? It goes hand in hand because remember I showed you this here. So when you have a rise in carbon dioxide, with water, carbonic acid is going to form and hydrogen is released. So a rise in carbon dioxide means an increase in hydrogen ion concentration. Do you see that? So an increase in CO2 also means an increase in hydrogen ion concentration. So they do go hand in hand. So as your hydrogen ion concentration increases, so does your CO2. So the same principle applies. So as there's an increase in CO2, more oxygen is going to get released. So the CO2, how does it do it? It binds to the amino terminus of the T state of hemoglobin, stabilizes that state. If it stabilizes the T state more than the R state, more oxygen is going to be in this. It's going to be the deoxy state, which means more oxygen is being released to the tissues. So this is the how. How does it do it question? Function. What happens because of it? So here we have three different graphs here. One with a pH of 7.4, no CO2. One with 7.2, no CO2, and one with both a combination of 7.2 and CO2. So this is really a double effect, right? So our pH, normal pH is at 7.4, so that's your normal. So that's going to be probably my 66%. I'm going to be talking about so much. So my pink curve will be 66% unloading. You know, as pH starts to decrease, the shift is more towards the right as more oxygen gets released. And that's what you see. 77%. So the last one is like an additive effect. Not only do you lower your pH, but you're increasing also the CO2 concentration. Right? So this here is an additive effect. So compared to the other one, with just the low pH, you're going to even see more oxygen being unloaded to the tissues, and that's what you see, 88%. All right? I would not ask you a question to compare this. Okay, I would not ask you a question that compares this. I would not ask you this. Okay? I would not ask you what would happen if I increase to 3 ppg and lower pH and have you compare it to what happens when you increase CO2 and decrease pH. because you need to know the concentration and in what concentration it affects more or less, right? Is it gonna be 78%, is it gonna be 79%? And I wouldn't ask you that. I would make it so that they are separated quite a bit like this, so you'll be able to tell the difference. Okay, so this is the example of that. Here's my purified, right? No 2,3 ppg, no CO2, it's just a normal stripped hemoglobin. So that would actually be more of a hyperbolic curve, like the pure one. So that would be a hyperbolic curve. And so as we increase CO2, see this I wouldn't ask you. I wouldn't ask you which curve would shift more towards the right. I wouldn't ask you that, okay? All I need you to know is if I add 2,3 ppg or CO2, my shift is going to be more towards the right because the T state is stabilized and more oxygen is getting released. But I can ask you to compare these two, right? Either or. I can ask you to compare what happens when I add both. What happens when I increase my 2,3 ppg and my CO2 compared to having one or the other. And you know, if you're adding more to it, then it's going to be an additive effect and it's going to push more towards oxygen release. And that's what you see with this last pink curve here. Here. Okay, so this is actually the blood sample that they're showing you that has that most oxygen released. Okay, so this is a summary of what we've talked about. When is T state stabilized? When is R state stabilized? So T state is stabilized when you have a rise in 2,3 ppg concentration, right here, and there's an increase in 2,3 ppg concentration. When you have an increase in CO2, you have an increase in CO2, more oxygen is going to get unloaded. Also, at the same time, going hand in hand, is your decreased pH. Okay, decreased pH, or in other words, high hydrogen ion concentration, right? I can say it either way. Right? I can say it either way. So any of those cases will stabilize my T state, and of course, the R state is the reverse. So when you're studying, first, of course, know when the T state is getting stabilized and when the R state is stabilized, and why. Why is this happening? How is it happening? And what's the significance of that in terms of oxygen unloading and my P50 value? So when we talk about CO2 and the hydrogen ions, this is called the Bohr effect. So the person named Bohr identified that what happens with hydrogen ions and CO2 in terms of release of oxygen. So this too, hydrogen ions and CO2 levels as it is related to hemoglobin is called the Bohr effect. Okay? That's the Bohr effect. Okay, so the last thing is, I guess, diseases. So we're going to look at sickle cell anemia as the disease. It is a molecular disease, it's genetic, so that means there's going to be a mutation. So sickle cell, we've got these little certain nice circular shaped cells, and then these folks with sickle cell will have this moon-shaped, half a moon-shaped, sickle-like. And so these sickle-like moon-shaped red blood cells will flow through and get trapped. And so the oxygen is going to deliver less. So in the beta chain, there's a mutation in the hemoglobin, specifically in the beta chain. And that mutation is here, shown in red. Okay, so in the normal, the beta gene, there's a glutamic acid. In the HBS, we call it HBS because it's a sickle cell anemia hemoglobin. That's what we refer to. Okay? So that's the term we refer to. So the hemoglobin S, that mutation is a valine. So what do you see the change? Thinking back to your amino acids, tell me what the change is going from a glutamic acid to a valine. Which one's non-polar? Okay. So you're going from one single change in the gene to make it go from a polar amino acid to a non-polar amino acid. And this one single change changes the conformation of that hemoglobin to make it sickle cell. Just one change. Okay, and this goes back to our gene. So when we really talk about the molecular going from the DNA to RNA to the protein, it's actually coming from the DNA sequence. There's a mutation in the DNA sequence, which then, of course, domino effect, right? When it gets transcribed to a messenger RNA, it's there. When you transcribe that messenger RNA and translate it into protein, that's what you're seeing. The change in that translation of that protein goes from glutamic acid to a valine, right? So that makes it sickle cell shape. Sickle cells rupture, right? Because they're really unstable, and they release iron because they rupture. And if you guys have any sort of a physiology course with Dr. Mockett yet, he probably talked about reactive oxygen species. And these oxygen species, reactive oxygen species, will break down lipid and protein. Okay, so these are lone pairs, and they are going to be breaking down lipids and proteins. So if there's an increase in iron, they're going to increase the rate of these reactive oxygen species, which break down lipids and proteins. Another thing is this disease. Because there is an increase in iron, I mean, obviously an increase in iron could be lots of different effects, right? Could be due to many sorts of disease effect. But one of the things is that it could be secondary hemochromatosis, and that's an overdose of iron. So if they do see this iron increase, it could be due to the fact that this person has sickle cell anemia. And this sickle cell anemia is causing an increase in iron, which causes this secondary disease. Right? Okay, so going back to our model here, this valine now, the glutamic acid was a polar, so it is exposed to the water area, right? Because it was polar. Now we have a mutation, now it's a valine. So it doesn't want to be in front of the water because it's non-polar and it's hydrophobic. So what happens is these hemoglobins, these red blood cells, will clump up together, really hiding it away. Really hiding away. You can see that here. It's trying to clump away and hiding it away through, again, non-covalent interactions. In this cartoon, you see it hiding away. This protrusion here is your valine. It tries to hide away from the water. And this sickling that you're seeing is what you're seeing with the sickle cell anemia, which causes that sickle-like shape. All due to that one change in that, making it into a hydrophobic amino acid. This is not something I want you to memorize, but I want you to see that causing a change in one amino acid have detrimental effects. And it's not just sickle cell anemia that's the disease, but you've got all these different diseases associated with it, right? You've got diseases where you see small amino acid going into a acidic amino acid. That's a big change. You can have something like phenylalanine going into a serine, again, hydrophobic, going to a hydrophilic, right? So any of these changes, don't memorize them, I'm just showing you, but you can look at them. It's a good practice to see, just by changing one amino acid and the properties that you've seen with the amino acids that I asked you to learn is now playing a role. Okay, I'm going to stop here. We'll talk about complete thalassemia next class and work on carbohydrates.